I'm not an oncologist. That's going to be a shock for today. I'm actually an ecologist. I deal with ecology. My background is broad and diverse and kind of messy. I started out my life looking at butterflies in Africa and then somehow spun into smaller bugs, looking at bacteria in Antarctica. And then in 2010, when I was offered a professorship at University of Chicago, I was sitting in my office one day, and which is just down the corridor in the Department of Ecology and Evolution at that time, and a surgeon walked into my office, and he said, "I've got this problem. I've got these bacteria that are hanging around in my patients' guts, and I think they may be killing my patients." And I said, "Oh, yeah, isn't that your job? You've got to get rid of the bacteria, stop them from killing your patients." And he was a very interesting guy, very, very engaged, and he alerted me to the fact that he needed help analyzing how the bacteria were interacting, how they were responding to their environment. And I sat down with him and I said, "Well, you do understand, I'm a marine biologist. I work in oceans and soils sometimes and plants, and not really in humans." And it very quickly became apparent from talking to him and then doing a bit of literature diving that there was no real difference. There are bacteria, there are viruses, there are fungi in every ecosystem on this planet, and they have the most profound impact on our world imaginable. And they have one of the most, I believe, profound impacts upon human physiology and human survival on this planet. It's only in the last 100 years that we've really been able to live in a world where we could eradicate some of the diseases that were so prevalent in the 19th century and before. And we've got very good at wiping out or vaccinating ourselves against those diseases that we've almost forgotten them. In fact, the rise of cancer as one of our major concerns and threats coincides extremely well with the reduction in the threat of diseases that we could easily vaccinate against or eradicate through the use of antibiotics. There's almost a perception in the public sphere that, I'm sure people were dying of cancer in the 19th century, but my God, tens of millions of people were dying of tuberculosis. And it was such a regular occurrence, we had a very different perspective of death back then. You know, we'd have pictures of our dead family members alongside us taken because we accepted death as a commonality. We don't accept that anymore, thankfully. We live in a wonderful world where we can have a big impact on the world. And I'm going to tell you a little bit about what I believe is a revolution in our understanding of how the combination of ecology and cancer therapies and psychotherapy and gastrointestinal health, wellness, atopy, asthma, HIV even, how these implications of ecology and medicine are coming together under a precision medicine forum. I was a member of the White House's precision medicine team to try and understand the impact of the potential impact of the application of microbiome science on our ability to actually perform a precision health diagnostic for individuals. And so I'm going to give you an overview of some of that work that we put forward in that place. But first of all, I'll tell you a story. I have a genetic mutation. We've heard about this. There are 23 to 24,000 human genes in the human genome. And one of my genes, APOE, has a double mutation, which gives me an extremely elevated risk of developing Alzheimer's. I got this from my mother. It's one of my mother's very wonderful gifts. And I passed it on to my eldest son. Luckily, my youngest son's missed it. My father doesn't have it, so we know it comes down that maternal lineage. And yet my mother's nearly 70 years old, and she doesn't have Alzheimer's. She has a very highly elevated risk factor, but has never developed the disease. Why is that? And we heard about people developing cancers, this idea of chance mutations, but there's also an idea of chance activation, that a genetic mutation doesn't necessarily lead to a disease. Okay, and why is that? Environment. Not the environment that Sid was talking about, but an environment more along the lines of an understanding of how the environment is affecting activation of a disease state. In this case, we're starting to find that Alzheimer's is very closely linked to changes in our microbiome and that the development of the disease and the persistence of the disease can actually be attenuated by changing the microbiome in our animal models. We've never been able to show this in a human yet, but we're working on that. But let's talk about that from this perspective. So I have a genetic mutation. I sincerely hope I don't come down with the disease. It'd be catastrophic if I did. But maybe there's some other component of my body that could be talking to that. We call that the microbiome. There's approximately 50 trillion bacterial cells in your body. There's around 30 to 35 trillion human cells, and about 50 trillion bacterial cells, so about 1 to 1.5 times as many bacterial cells. And they have around 7 to 8 million genes in their genome. We call that a collective genome. There's a huge amount of genetic material inside you that isn't you. And you don't escape from that. That's pervasive. That's part of your life history. We get that microbiome from our mother. It weighs around 3 pounds by the time you're an adult, but very early in life, when the amniotic sac ruptures, you are essentially sterile. And then you acquire microbes through your transition through the vaginal cavity or via other delivery mechanisms. So, for example, in this diagram, we have these dots. Each dot represents a microbial community taken from an individual. Essentially, we take a cotton swab, we swab a piece of the microbiome, we sequence the genetic information, we identify the species, bing, bam, boom, you have a profile. Up here, you can see on the top, these yellow dots represent the microbiome profile from the mother's oral cavity, from her mouth. And the dots down here either represent the mother's vaginal cavity or mother's skin. And then babies born via a cesarean section delivery or via vaginal delivery. These are the microbial starting cultures, like sourdough bread, right, or brewing culture. It's kind of, weird analogy, but you get the idea. This is what you start with. Your vaginal cavity delivery is the microbiome which you evolved to start with, right? We've evolved in a sea of microorganisms. All life has. Microbes have been around for 4 billion years. Multicellular, large life has really only been around for around 600,000. So they changed the planet, and then we could evolve, right? And we've evolved alongside them. This microbiome is the one that you would have got had you been born in Chicago 200, 300 years ago on a prairie. You would have got the vaginal microbiome, and its contents may actually be affecting how your body develops. Children that are born via C-section, in very large epidemiological cohort studies, children born via C-section have an elevated risk for a number of atopic diseases, asthma, food allergies, even some neurological diseases such as autism and cognitive displacement. We don't fundamentally understand why that is, but there is a suggestion that this immediate colonization by a differential microbiota could be a factor contributing to that development. In fact, some people are now suggesting that you should do a vaginal seeding. So you take a gauze or a tampon, you place it in the vagina during birth, and then you actually use that to swab the baby after C-section, right? And many physicians would say that's extremely dangerous. You don't know what kind of bacteria you're rubbing all over that child. I'd say most women know pretty carefully what bacteria they're rubbing over that child. But there's actually a surge in movement towards this kind of activity. I do not condone it. I just don't oppose it. But either way, this leads to us being microbially unique. Each one of you is microbially unique. I actually work with the Department of Justice on developing the microbiome as a forensic tool, one of my many other side projects. And I could identify every single one of you based on the microbial signature you've left behind on your chair right now. At a crude level, I could identify men versus women very quickly for obvious reasons. These pants do not stop bacteria, right? This is just like fishnet stockings to microorganisms, okay? In fact, you are a cloud of microbes, as I'll show in a minute. But this microbial community is extremely important in helping us to identify the processes which lead to certain disease types. So, for example, even identical twins have a unique microbiome. They progress through life in a very unique way. We each are exposed to the environment in a very different way as we grow up, no matter what kind of clothes our mother dresses us in. We may be licked by a dog or eat too much of a different type of dirt or fall down in a different way, and that changes the types of microbes you're exposed to, which changes your immune system development, which changes the progression of succession, ecological succession of your microbiome. So we end up in this microbially unique space. But over the last 100 or so years, we have been eroding that microbiome. The rainforest ecology of our body has been under attack for very good reason. We suffered at the hands of microbes for many tens of thousands of years as a species. We were at their beck and call. We died in our millions. In fact, most of you would not be alive today had your parents and grandparents not been given an opportunity to live in a world of vaccines and antibiotics. So that erosion, all while it's kept us alive, has also led to, we believe, a profusion of other disease types, chronic diseases, many of them, which have led to a shift in our health status. And atopy, asthma and allergies, conditions that we wouldn't normally have seen in our population appear to be increasing. And we think that this erosion may be a significant proportion of it. In fact, there are currently 399 ongoing clinical trials registered by the US government exploring the influence of the microbiome in conditions such as you see up here on the screen. This is a changing field. It's a very, very early stage field. I am not able to show any of the kind of clinical data that Sid showed, but I can definitely state that we are in a process of trying to understand the implications of this in a very rigorous and trialed way. We know that the microbiome affects us in various stages. Here's some neat examples. Whether a mosquito will bite you and where it will bite you on your body can be determined by the types of bacteria you have in your skin. Working with the Department of Defense, colleagues of mine have been able to demonstrate that the bacteria living on your wrist, for example, metabolize your sweat, release volatile compounds which are sensed by the mosquitoes, and that attracts them to where they land. We can use this information to try and interrupt that signaling relationship between the bacteria and the mosquito to reduce diseases caused by the spread through biting flies. Tylenol, for example, acetaminophen, can be metabolized in certain ways by bacteria living in the intestine, which can lead to long-term liver damage after a prolonged period of time. Trying to be able to identify that could be useful. That's just one example. There are many drugs that are either deliberately, such as prodrugs, metabolized by the microbiome to have an effect, or undeliberately or inappropriately or inadvertently metabolized by the microbiome to have a less desirable effect. Yes, that is two flies involved in some kind of romantic endeavor. But this is an important side effect as well. We know that the bacteria in those flies can be useful in changing their behavior. In fact, I can take the microbiome from one fruit fly, transpose it into another fruit fly. This is where it gets complicated. And I can make the second fruit fly fancy the fly that the first fruit fly fancied. Did you get that? Yeah? Try saying that after 2 glasses of wine. It doesn't work. Not that I've had 2 glasses of wine already today, you understand. That would be weird. But this is important. I can change the behavior, mating behavior or whatever, of an animal by altering just its microbiome. And we are starting to see in many of our studies that by understanding the microbial community in an organism, such as a human, we're actually able to discern differences between people that suffer from neurological disease. And that's why putting the microbiome in your medical records and having it as a potential component of a precision medicine program, such as the Precision Medicine Initiative, is, in my view, extremely important. Here's one of the few well-performed double-blind clinical trials that have been developed for microbiome therapy. It's a fecal microbiome transplant therapy. This is where we take the microbiome of a healthy individual in their stool and literally transplant it wholesale into the colon of somebody who has a Clostridium difficile infection or persistent C. diff infection. This is actually not a new therapy. The Chinese have been doing this for 1,600 years. They called it yellow soup. They would take the stool of an infant, a healthy infant, and mix it up in a broth and feed it to somebody with gastrointestinal sickness. And in many instances, it's been shown to, in the ancient Chinese medical literature, been shown to be effective in treating disease. And in this case, it was highly effective. The standard mechanism, The standard mechanism for treating Clostridium difficile infection was vancomycin treatment, antibiotic, a non-absorbable antibiotic. And you see we get about a 30% accuracy rate in terms of recovery. When we do a fecal microbiome transplant, that jumps to 94%. In fact, this clinical trial was ended early because it would have been unethical to continue treating the other individuals who weren't getting the fecal microbiome transplant. So that's a very positive outcome. Many times, however, subsequently, fecal microbiome transplants haven't been shown to be a magic bullet for many other diseases. It's not as effective. We don't fundamentally understand why these transplanting this 1,000 bacterial species from one person to another should change the disease pattern. We know it's very important for immune system activation, and we're starting to understand that by looking at children and how their immune systems develop. And for example, our interaction with the environment can be a key by moving from living outdoors, where we are exposed to a rich, diverse microbiome, to indoors, where the microbiome can be much less diverse, and through the use of cleaning products, antibiotics, etc., which kill the good bacteria along with the bad, we may be inadvertently reducing the diversity of our microbiome, leading to us missing some of our microbes, which could be affecting our immune system and leading to the development of allergies, even seasonal allergies. We think that by adding bacteria back into the microbiome of our body, the correct kinds of bacteria, specific types, we may be able to protect children from developing some of these conditions. We think children are particularly susceptible because their microbiome is still developing up until about the age of 3. So by getting in while the microbiome is still in flux, before the ecosystem is settled, we can have an effect on developing it. Any good gardener will know that in order to build a good garden, a good healthy ecosystem in your garden, you need to get in there early and have an effect on it. Starting to tackle an already very well-developed garden is very, very hard to push it in a new direction. So we're working as caretakers of the microbiome at trying to get in there early and figure out how to address it. And some work we published in New England Journal of Medicine a few weeks ago has demonstrated this highly effectively in infants growing up on farms. We work with Amish populations in northern Indiana and Hutterite populations in North Dakota. Both these populations are from the same genetic lineage in Eastern Europe, both from very agrarian societies. And their microbial exposure in those agrarian societies was always richly affected by the animals they worked with. Now, these two populations came over here to the US. They both eschewed technology in the same kind of way as we would traditionally accept for the Amish. But the Hutterites live a very different farm life to the Amish. The Amish live on personal family farms. The children muck in very early in life. There's a little girl who's been helping her dad to actually harness up the horses. These are 1.5-ton Shire horses, incredible animals. And that's how he plows his field, right? But the children work. I actually take my kids to this farm about twice a year to take samples from the people and to show my children, right, that children can exist without an iPad. It's technically possible. And they're blown away. I'm also blown away. But on the Hutterite farms, they actually adapted a very technologically advanced farming system. At home, they live in a technology-free religious existence. But on the farm, children aren't allowed on the farm because it's too technologically advanced and there's health and safety regulations. So the children in the Hutterite families aren't exposed to the same kind of microbial exposure that they are in the Amish families. And if we take the dust from the bedrooms of those Amish children and expose animals to that dust, literally by putting it inside their intestine, the dust activates an immune response which stops those animals from developing asthma or wheezing phenotype. But if I take the dust from the Hutterite children's homes and their bedrooms, I do not see the same kind of response. But if I take the dust from the barns of both of those communities, the barns where the animals are living, I can stop the mice in our experiments from developing asthma. And we know that children growing up with a dog in their home under the age of 1, just a dog, have a 13% reduction in the likelihood of developing asthma. Children growing up on a farm generally, not just an Amish farm where they're actually working hard at living there alongside animals, but a farm generally, have a 50% reduction in the likelihood of developing asthma. This is the hygiene hypothesis. The idea that by moving away from the environment, we are somehow eradicating or breaking a link with our ecosystem. And that's changing the way our immune system develops and leading to shifts overall in our body's homeostasis. We've done this with food allergies, working with peanut allergies and with milk allergies in children to try and identify ways of adding the right kinds of bacteria back in order to allow those children to live a more healthy life. In our animal models, we can take over here on the graph, we can show that an animal, a mouse growing up with a rich contextual microbiota, our SPF mouse, has very little kind of allergic response. Its IgE response, its immunoglobulin E response in its serum, is very low when we actually challenge it with peanut allergen. If I take all the bacteria away from that mouse, I make it germ-free, its allergic response to peanut allergen is off the charts, goes through the roof. If I reconventionalize a germ-free animal, if I add bacteria back into it, I can bring that response back down and see the blue graph saying comb. That's conventionalized animal. If I add in just Bacteroides uniformis, one type of bacteria, I don't see that rescuing of allergic infection. If I add in Clostridia, a type of bacteria that produces a compound called butyrate, butyrate is a food for our T regulatory cells in our immune system of our gut and helps to regulate how that immune system responds to inflammation, then we see a significant recovery. And we've started to develop this for cow's milk allergy, performing a study in infants. By adding certain bacteria into the experience of those infants under the age of 6 months, we were able to recover almost 50 to 60% of those children from having a cow's milk allergy. And by tailoring and developing those probiotics, and these aren't the kind of probiotics you can buy in Whole Foods, but very specific clinically relevant probiotics, to very specific elements of that disease for those individual children, we're able to actually increase that in our early stage animal models significantly higher, up to 70 to 80% recovery. And we're hoping to start a clinical trial on that very soon. Depression, anxiety, I said we can change the mating preference of a fly, but I can also look at how people respond to their depression in a microbiological factor. We know that people change their neurological status with diet when they move away from living where they were previously. We've seen this quite regularly. But we're starting to see that bacteria in the gut who produce some of our neurotransmitters that are very important for the maintenance of our central nervous system neurological function, when they are disturbed, we see significant shifts in the propensity towards certain types of neurological phenotype. Autism has been shown in this, in both animals and in humans, that when we alter the microbiome, we can alter the type of autism phenotype we're developing. With depression, we're starting to see this in our perinatal and postnatal depression cohorts at University of Chicago. We're starting to see a significant shift in that space. And we actually work very closely with University of Illinois Chicago on that study. We also have groups that are looking at other disease types, such as cognitive development. We have a new study. We're working in the south of Chicago. I'm in Northwestern in the north, University of California, San Diego down in La Jolla, to look at how children develop after they leave the hospital under conditions of either poverty or affluence. And we believe that the microbiome in children under poverty is significantly disturbed. Based on our existing evidence, this is highly effective. And then we follow them over time and see how they develop up until school age. This is extremely exciting research, heavily funded by National Institutes of Health and various other agencies. We believe, and we can show this, that I can take the microbiome from a depressed person and put it into a mouse and cause a significant shift in that animal's neurological capabilities. So this is something that's causative, but we don't understand how it's causative. The mechanism is very, very difficult to discern. We can see here a great study where colleagues of mine took an animal, a mouse here, and a normal mouse stays hidden inside that box quite happily. If we take that box, that animal, and we remove all this bacteria, it will run around as if it hasn't got a care in the world. It loses all of its anxiety that would normally keep it alive in the wild, and it gets to remain hidden, okay? The genetics behind this are not the mouse's. Its behavior is being altered by the type of bacteria that's present there. In the absence of bacteria, it no longer has a keep hidden response. It will walk quite happily out into the wild and probably be eaten by the first animal that sees it. Obesity is affected by the microbiome. A morbidly obese man weighed 385 pounds, lost 113 pounds after 9 months of living on a whole grain diet. I do not recommend it. He checked his microbiome before and afterwards. And there was one bacteria, Enterobacter cloacae B29, that when he cultured up and grew up and placed into a germ-free mouse animal, there was a significant change in its morphological phenotype. Put on an awful lot of extra white fat, okay? And we don't fundamentally understand, again, the mechanisms, although work with Eugene Chang and myself at University of Chicago started to elucidate some of these components, but we do see a causative relationship. The presence of these bacteria causes a weight gain. Certain bacteria, when we add them in, actually can protect against weight gain. And a germ-free animal will have to eat and eat and eat all day long with no diurnal rhythm because it can't put on any weight, and it's constantly hungry, and we don't fundamentally understand why. Bacteria seem to regulate the behavioral response for appetite, as well as how the energy is processed and utilized in the body. In the room you're sitting in right now, this built environment, this ecosystem that we've created, you're living in a microbial soup. Each one of you is emitting 36 million bacterial cells into your immediate vicinity. Think Pigpen from the Peanuts cartoon, right? You have this dust cloud around you. And you're each becoming more microbiologically similar because of that proximity that you're interacting with. The people sitting closer together are becoming more similar, and the people sitting further apart are not having that kind of interaction. Your interaction with your environment doesn't stop at your skin, just the same as it didn't stop at your outside of your clothes. You are constantly interacting with your world through your microbiome. And we are working with architects and building designers and urban planners to try and understand how our interaction with the microbiological world is affected by this new ecosystem we call our homes, our offices, our hospitals. And this has become extremely important to our understanding of microbiological health. How does the world in which we've chosen to live affect our acquisition and development of our microbiome? Think if I take a baby and I place it into a world where there's only skin bacteria emitted by human beings. How's its immune system going to respond to that environment? This is one such world. I brought my infant child into a home which didn't have any plants, any animals. It just had me and my OCD to keep the house clean. You know, my house was very clean. And my microbial interaction, each one of these lines represents a directionality for a bacteria moving from one surface to another, for either the feet, the nose, the hands of myself and my family, our floors and our surfaces in our home. We were able to see that children being brought up in that environment without any other kinds of sources had a very strict interaction. It was basically they were acquiring skin bacteria from their fellow human beings. And through work from many groups around the US and in Europe, we've shown that that skin bacterial interaction doesn't elicit the same immune response as, say, interactions with dogs. And I said that dogs in the home will decrease your likelihood of developing asthma if you have it under the age of 1. If you get it later and you've already developed asthma, dogs can be a trigger for your asthmatic attacks, so go figure. But early on, when you get that early immune response, the dog bacteria can play a significant role. And we found out from work at University of California, San Francisco with Susan Lynch that certain types of bacteria from dog feces, when placed inside an animal, a mouse, will significantly reduce the likelihood of it developing an asthmatic attack. And here we're seeing a map of dogs interacting with the people in a home. And this interaction space was much more diverse. Couples living with a dog are always more microbiologically similar than couples living without a dog, controlling for all other variables, okay? So actually having a dog where the dog's promiscuous, it loves both mummy and daddy, and like spreads the microbiological love around, significantly affects the degree of microbial sharing. All of these are important environmental consequences, things we tend to ignore when we just consider the human being. We use this information, or my wife used this information, to bribe me into rescuing a dog from Kentucky. His name is Captain Bo Digley, and he now lives in our house. We can also, I said, use this for forensics. I can track each one of you with my nefarious database. Here we have a young man, person 1, living with his mum and dad in a home with 3 dogs. You can see person 3 is the mother, and on the kitchen counter, her microbiome over the 6 weeks of observation constitute nearly 100% of the bacteria found on that surface, until she goes and lives with her sister for a short period of time, and then the father's microbiome takes over. So we can even track gender bias in a home using the microbiome. And we're using this to track dangerous break-ins and burglaries around the US in a partnership with police departments in about 17 cities. Just to very briefly touch on the hospital microbiome project, we've sequenced the microbiome of many different environments, tracked it over time, saw how it is varied and affected by different treatment strategies, different places. And one thing we found was that the microbial community exchange that people have with their environment had very little impact on whether they developed an infection. And in fact, many of the infections that we saw people developing in a hospital actually came in with the patient, okay? And so they already had the bacterium that caused their disease when they arrived. And we've used this in surgical infections to try and understand a paradigm of why surgical infections cause a disease. And this is my very last slide, so I apologize for running over a couple of seconds. So this is a surgical gut. When we want to remove a carcinoma, a polyp, for example, we remove a section of the gut, and then we do two things in that instance. We flood the gut with oxygen, which kills off certain bacteria and inactivates others, because bacteria in the gut don't like oxygen. We pump the body full of antibiotics. That also has a negative effect on many of the bacteria. And then we join the gut back up during our surgical practice, and we create an anastomotic junction. This does one last thing. The body is under stress, sucks nutrients out of the gut cavity, causing the bacteria to suffer nutrient stress, and they relinquish a new phenotype where they jump onto the gut wall. In doing that, they release a new series of enzymes which actually start to break down the gut wall and cause it to open up. These are commensal organisms living inside the gastrointestinal tract that are sensing their environment. They're living organisms, right? Like wild beasts living inside you. They're sensing their ecosystem. When they sense that nutrient depletion, they do the only thing every other organism in the world, including human beings, does when it starts to starve, and that's go after the source of nutrients. And the source of nutrients is this lovely bit of exposed tissue in the gut. They get there, they release celluloses, they release enzymes which start to degrade the connective tissue in the gut lining. That releases phosphate and nitrogen, which they can use for food, and they break open the gut, causing a massive septic shock and often death. We found that by adding controlled nutrient sources into the gut of animals undergoing surgery, we can alleviate this likelihood, and in those instances turn almost 80% mortality in our animal population back to 0%. So we think that by affecting change in the microbial ecosystem of the gut, we may be able to help treat diseases in the hospital in a much more rigorous way. And I'll just leave it there. We have a microbiome center at University of Chicago, Argonne National Laboratory, which is the local federal lab, and the Marine Biological Laboratory out at Woods Hole, which the University of Chicago monitors. And we have relationships with all of the major universities here, covering everything from marine and environmental microbiological research all the way through to human health. Thank you very much.